Hey guys, so now that we've got the cellular toolbox, we can go through all these cell parts and kind of see how those tools are used to make these parts function properly. A lot of this will be a review. We'll just kind of rip through the parts. It's not the kind of thing AP is going to ask you about. They're not going to ask you what's the function of whatever. You're going to have to know what cells are capable of and how they do things, but not necessarily the names of parts. But we'll go through real quick. First, and probably most important, is the nucleus. And the nucleus in a eukaryotic cell is just a place to put the DNA and keep it safe. DNA is inside of there, away from all the cellular functions that may cause, damage to the DNA. It's just another way to mitigate that mutation rate. For multicellular organisms, a high mutation rate is not beneficial. We have to have some mutation so that we can have genetic diversity, but we need to protect ourselves from genetic disorders and cancers and things like that. So the nucleus helps us do that, helps keep the DNA safe, keeps it away from all the rest of that activity in the cytoplasm. In the nucleus, there's a nucleolus. This is where ribosomes are made. So maybe worth knowing that ribosomes are made in the nucleus. It helps us also kind of point out that ribosomes are not a location. Ribosomes are everywhere. So inside the cell, you'll have ribosomes all over in the cytoplasm. Some will be bound to the endoplasmic reticulum, and some are still in the nucleus because that's where they get made. So you can't say to the ribosome as if that's a location. It's not a location. But all the nucleolus does is makes ribosomes. Ribosomes, of course, are made up of some RNA strands and some protein strands. So they're a big enzyme complex. It's a big protein RNA complex that uses an mRNA recipe to make proteins, to assemble chains of amino acids, to make polypeptides, right? It does that by following that mRNA code. The mRNA code, of course, came from the DNA. The RNAs you find in a ribosome are called ribosomal RNAs, or usually just rRNAs. The genes that code for these, they're very similar in every organism on Earth. If you look at the sequences of the DNA that have these RNA codes, they are very, very similar. And there's a reason for that, of course, and I'm sure you can identify it. This is one of those things that's highly conserved because if you mess with it, you mess with your ability to be alive. What's also interesting is that there are many, many copies of the ribosomal RNA genes in your genome. So in your DNA, there are several places in your genome that have copies of these genes so that if you get a mutation in one of them, you're still making plenty of ribosomal RNA. There are slight differences in the sequences. And when they ask what pattern do you expect to see, well, the organisms that have a more recent common ancestor are going to have fewer of those differences, right? And the ones with a more distant common ancestor are going to have more of those differences because they've had more time to accumulate differences over time. Now, again, we're not going to accumulate differences that affect the function, but we will accumulate differences in parts of the sequence that don't really affect the function when they get changed. So the rough endoplasmic reticulum, this is where we build proteins that are going to be secreted outside the cell. This is also where we build proteins that are going to be membrane proteins. And we talked a little bit about that when we looked at the secretory pathway, when we were talking about vesicles and things like that in the last PowerPoint. We'll cover that a lot more later on, this whole endomembrane system. So the proteins that are made in the rough ER, those end up either secreted outside the cell or embedded in the cell membrane. This is where that starts. The ribosome is going to grab the mRNA. It's going to assemble the first few amino acids into a polypeptide. And that sequence of the first few amino acids will tell the ribosome, hey, stop right there. It's actually another protein that gets involved. Stop right there. Don't translate this. Don't build this protein until you get to the ER and then build it in there. And so the ribosome will go to the ER and build that protein into there. And what you can see, like both these images, you can see there are tons of little ribosomes studded on the membrane of the ER. And it's highly folded because we need this massive surface area so that we can build all these proteins. Both these slides are probably from cells that secrete a lot. I know the one on the right here is from the pancreatic cell. I think the one on the left is as well. But if you look at these cells, they are just packed with ER because that's all they do is secrete proteins. So when we ask a question like which cells have a lot of ER, and this is a kind of question AP likes to ask, they want you to know what cells, what tissues are going to be using what organelles. And so any cell that secretes stuff, mammary gland cells, pancreatic cells, glandular tissue that produces protein hormones, these are all going to be packed with ER that is studded with ribosomes, right? That's what makes it look rough in the microscope. It's just all those ribosomes. But that rough ER is going to be packed into those cells that are secreting things. The smooth ER is one that we don't have to know too much about. Its basic function, though, is making lipids, and it does some detox stuff. Got a little exclusion statement from the AP people that says basically you don't need to know too much about the smooth ER, just it makes lipids and it's used to help break down some, toxic chemicals that may end up in your cells. When you did your cell project in freshman year, when you said the cell is like whatever, a skateboard factory or an iPhone or Hogwarts or whatever it is you made it, it's a common project freshman bio teachers do. I'm not really sure why, because a cell is not like any of those things. Maybe it's a little bit like a skateboard factory, but the shipping department was always, the Golgi apparatus. I think in your Hogwarts analogy, it was probably the owlry was the Golgi apparatus. But it's basically prepares things for leaving the cell. So we build those proteins at the rough ER. They get transported by a vesicle to the Golgi. The Golgi is then going to make some final tweaks and adjustments that kind of help determine where they're going to end up, or maybe help just get to their final shape. A lot of times we're talking like glycoproteins, which are proteins that are embedded in the membrane, have chains of sugars on them. Those chains of sugars will be assembled there in the Golgi. But we're going to then be able to pass those onto the cell membrane. So it's just kind of a final processing place for anything leaving the cell. Also for anything going to another organelle. Like this is the way we make lysosomes. We build these proteins in the ER. We make some adjustments to them in the Golgi, and then bud off the Golgi, and we have a lysosome. We'll talk more about those here in a second. But it's just kind of a processing place along the assembly line that is that, endomembrane system. Cells that have lots of Golgi are going to be the same ones that have lots of rough ER because they're part of that same pathway. I'm running out of places to put myself. I feel like I keep jumping around in here. So lysosomes, these are digestive organelles. They are little pockets that we keep low pH in. We keep enzymes that are digestive enzymes inside of there. You don't want the stuff in there to get out, and because it would be very damaging to all the rest of your inside of your cell. And so we make these in the Golgi. We embed proteins into the membrane, and then we bud off of the Golgi. And those proteins are not active. Those digestive proteins, they're inside. The digestive proteins are maybe in the membrane, and there are proton pumps in the membrane. As the lysosome buds off of there, those proton pumps get activated, and those proton pumps lower the pH inside of the lysosome. And that activates some of those digestive enzymes. So they're zymogens. Zymogens are proteins that are only active in certain circumstances. And usually they're digestive proteins, because we don't want to just digest all the proteins that are in your cell. You want to be able to make these digestive enzymes, but not have them digest the cell itself. So you make it so that they can't be activated until they're in a specific location. Pepsin in your stomach is activated by hydrochloric acid in much the same way. You dump out pepsinogen, which is totally inactive, doesn't do anything. And then when it gets into the acid, it becomes pepsin, and now it starts chopping up proteins. So it doesn't chop up the proteins inside your cell, just the ones in your stomach. The enzymes we put in the lysosome work the same way. They are zymogens. Mitochondria, we're pretty familiar with these now because we've done cellular respiration. So mitochondria, their job, of course, is that they are it's kind of like a powerhouse of the cell. They generate ATP in a process that you know very well from last semester. If you look at the two membranes, the inner membrane is very, very foldy, right? All of those little cristae or those little folds, and that's all about surface area. And if you remember from cellular respiration, that inner membrane is full of electron transport chain proteins, right? So that's where we're doing all that pumping of protons. That's also where the ATP synthase is embedded. So more membrane means more places to make ATP. It is interesting to note that mitochondria, you don't make new mitochondria when you're doing normal cell division. Before the cell divides, you go through what's called G2 phase. You're going to make more Golgi. You're going to make more ER. You're going to have the DNA doubled. You're going to have everything, 2 copies of it, or enough for 2 cells of it, and you're going to split into 2 cells. But mitochondria don't work that way. Chloroplasts don't either. Mitochondria can tell when a cell doesn't have much mitochondria and will reproduce on their own, and it has nothing to do with the rest of the cell cycle. And the way they divide is they basically shove some of their DNA to both sides, and they have multiple copies of the mitochondrial chromosome in there. And they'll split inside and then split the outer membrane. The way they divide is almost exactly binary fission, which is how bacteria divide. Their reproduction process is essentially the same as a typical bacteria. And this is one of those clues to the origin of mitochondria. And we'll talk a lot more about the endosymbiont theory later on. So almost all eukaryotic cells have mitochondria. In each cell, there's usually a couple of dozen or more, depends on the cell type. The demand for ATP that a cell has, that's what determines how many mitochondria are going to be in there. So muscle cells have bajillions of these things. Neurons have tons and tons of mitochondria. Other cells that don't have the high demand of ATP are going to have fewer mitochondria. The two biggies in humans are definitely muscles and neurons. Mitochondria and chloroplasts have some interesting similarities. They have their own DNA inside of there. Their DNA is circular. They have a double membrane. They have their own ribosomes, which are bacterial ribosomes. So they're very different from most of the other organelles. Having their own DNA is really weird. Most organelles don't have DNA, don't have ribosomes, don't have a double membrane. Right? All of those are clues to the origin of mitochondria and chloroplasts. And of course, mitochondria are everywhere. They're in all the eukaryotic cells. Don't forget that they also exist in plant cells. It's easy to fall into that old misconception that plants do chloroplasts and we do mitochondria, and they're giving each other the things they need, right? The plants are doing it on their own. They're fine, and they're doing it for themselves. And we just come along as animals and grind up their babies and eat them. So speaking of chloroplasts are plant organelles. There's a whole group of plant organelles called plastids, and they are just basically membrane-bound pockets where they can put things that provide a certain function. And they sometimes are colored, so like what makes a fruit red, or an apple red, or a flower pink, or yellow, or orange, or whatever. Those are pigments that are stored inside of little, what are called chromoplasts. So the word root chromo just means colored. Chloro, the word root means green. So plast is just like body or shape. So chromoplasts are the colorful bodies or shapes, and chloroplasts are the green bodies or shapes. Amyloplasts, amylo is starch. So these are just starch storage. This is what you find in potatoes. Lots and lots of big storage compartments for starch. Those are amyloplasts. Chloroplasts, of course, their job is photosynthesis. We're pretty good at this. We know in the stroma, we have the Calvin cycle. We have the thylakoid membrane, where the electron transport chain is with chlorophyll that can excite those electrons, generating ATP and NADPH. All that stuff we've talked about before. But their job is photosynthesis. So this is Lynn Margulis, and she was the first to propose this idea that mitochondria and chloroplasts are endosymbionts. And technically the idea is older than that, but she was the first one to really kind of put together some of the research that kind of demonstrated why we think these things are this way. Why do we think that mitochondria and chloroplasts are not a native part of the eukaryotic cell? Why do we think they're an endosymbiont? And so she looked at all these things, like they have their own ribosomes. And if you look at their ribosomes, the sequences, they don't match the ones produced by your Nucleolus, right? They are separate ribosomes that are more like bacterial ribosomes than they are like yours. The DNA inside of bacteria, prokaryotic DNA is rings. There's no ends. Our DNA is long strands with two ends. Right? If you look at the DNA of the chloroplasts or the mitochondria, they're ring-shaped. If you look at the DNA of bacteria, they are ring-shaped. These are bacterial chromosomes that we find in mitochondria and chloroplasts. And the genes are bacterial too. If you took the DNA from your mitochondria and ran it in a gel, did a DNA fingerprint of your DNA from your nucleus, the bacterial DNA from any bacteria you want to pick, especially ones, though, that are like aerobic bacteria, and the DNA of your mitochondria, the mitochondria would much more closely match the genetic fingerprint of an aerobic bacterium than it would be your nuclear. They would be very different from your nuclear DNA. The genes are bacterial genes. The chromosomes are bacterial chromosomes. The mitochondria has a double membrane. They have their own ribosomes. They build their own proteins using their own ribosomes. So she took all of these ideas and started looking at them, and started also looking at the genetics of these things. And she proposed this idea of these endosymbionts, of mitochondria and chloroplasts were once free-living organisms that were engulfed by a larger eukaryote, and both organisms benefited. The larger organism got a steady source of power. The smaller organism got a more stable environment to live inside. Plus, the larger eukaryotic cell has more access to the nutrients that the mitochondria are going to need. So basically, the endosymbiont theory states that the mitochondria and chloroplasts were once free-living. They were engulfed by an ancestral eukaryote. They're endosymbionts. Endo means within, symbiote, symbiosis, right, living closely together. We often say that the double membrane is evidence of this endosymbiosis. Let me show you what I mean by that. This is like one of the few cartoons I ever made. It's terrible, and I apologize, but we'll watch it anyway. So this little guy here, that's the aerobic bacteria. This is the large eukaryote. And as it goes to engulf it, I have to shove the camera closer. That's how we zoom in back in the day. It surrounds it with a vesicle, so it does endocytosis, and then the inner membrane eventually evolves to be much more convoluted. This is really way too long. Had to fix that in. I apologize for that. So we still have the free living versions of these guys out there. We have mitochondria, and then we have aerobic bacteria that still live around out there, and they have these infoldings of their membrane that they do this electron transport chain in. They are able to use oxygen and generate a lot more ATP than a typical bacterium. Chloroplasts, we have cyanobacteria all over the world. In fact, we even have some eukaryotes that will live in like a pond, and they'll cruise around and eat stuff. And if they happen to eat the right kind of cyanobacteria, then they will swim up to the top and they will sit there at the surface of the water and use that cyanobacteria as a chloroplast. So they become a very plant-like protist. And then when they go to divide, one of them gets the chloroplast and the other one doesn't. And the one that doesn't has to go around and start foraging and eating. But it will just eat whatever it gets until it engulfs one of that same species of cyanobacteria. And then it will move up to the top and do the same thing and become a plant-like protist. So not only do we know this happened, we know that it still happens. We can see it happening all the time. I don't know that we've found a current version of the aerobic bacterium being engulfed by a eukaryote, but still, I mean, we've got all of this evidence, all these different types of evidence showing that these guys did not originate from our DNA. It didn't come from our cells. I think you could probably do this again, but go ahead and pause the video just to make sure. I'll wait. This is one of the most epic high fives ever, I think. I have no idea what it is. So here's endosymbiont theory, basically in one little timeline. You can see we've got this early prokaryote that was ancestral, but we get this infolding of the membrane to get the ER, the Golgi, the nuclear membrane. What's interesting is that the nuclear membrane is actually a double membrane. So you can kind of see how we get this infolding becomes the nuclear membrane here. It's also, so it's a double membrane, but it's also continuous with the endoplasmic reticulum. So the space in between the two nuclear membranes, that space is also the inside of the ER. It's the same space. So kind of interesting. You can kind of see that it's all from the same original infolding. So then we engulf, right, this prokaryote that could do cellular respiration. Then some of those engulf a cyanobacteria. These guys go on to become funguses and animals, and these guys go on to become plants and algae. So we have what are called vacuoles inside of cells. A lot of different cells have this. You have white blood cells that form a food vacuole, basically when they engulf a bacterium or something that's in your blood or in your body that should be digested. We'll talk about contractile vacuoles when we talk about osmosis later on. Basically, these guys live in fresh water, and so they are constantly dealing with an influx of water. So they will actually put it all in one spot and then squeeze it out physically. But these are just like a place to put something. It's basically a bag inside the cell to store something. In plants, we have lots of different vacuoles. Generally, plant cells have one great big vacuole right in the middle. We call it the central vacuole. It has a membrane around it called the tonoplast. You don't really need to know that, but it's basically a place to store a lot of different things. In fact, if you remember when we did the catechol and polyphenol oxidase day-to-day with the graphs where we were looking at enzyme activity, I told you that the catechol is stored inside the central vacuole. The polyphenol oxidase is in the cytoplasm, and they only come together when the cell is damaged, right? So that they're separated just by putting it in that central vacuole. One of the main functions of a plant vacuole is to provide turgor pressure. It's a place where they can put all kinds of molecules that are soluble in water and also just salts. And so all of that causes osmosis to occur, which we'll talk a little bit more about later, but water goes rushing into that thing and that swells up. And so it's always pulling water from, the xylem, which is a source of water that comes, connects all the way down to the roots. But it pulls this water in and it makes the cells swell up. And that's what makes the cells, full of water, full of turgor pressure, and gives the whole plant some structure. There are also microbodies or other little organelles that may be in certain cells, in different organisms, in different parts of the body, whatever. Peroxisomes, we have these glycoxysomes. These are all different things you're going to encounter. If you ever need to know something about one of these for AP, they're going to tell you about it. There are lots of weird and obscure little organelles that they could bring up, and it's not really possible to teach you all of them. So if you need to know the function, they'll tell you the function, and the cellular toolbox will help you figure out how it does that function. We need to talk about the cytoskeleton too. It basically is what gives cells, especially animal cells, their shape. Plant cells, it's largely turgor pressure inside of that, cellulose cell wall. That's what gives the cell its shape. But in animal cells, that shape is almost entirely due to cytoskeleton and then membrane proteins. So we have two main parts of the cytoskeleton. We have microtubules, which are kind of central, irradiate out central from centrioles, radiate out in the middle of the cell. And then we have actin fibers, which are typically around the outside, just inside the cell membrane. We mentioned these before when we talked about motor proteins. Motor proteins always walk on cytoskeleton, whether it be microtubules or actin fibers. Another word for actin fibers is microfilaments. Most of the time you hear actin, though. This is a pretty cool image where they've labeled with fluorescent dyes DNA, and so that's why you see the nuclei in the blue. The actin fibers are stained in red, and the microtubules are stained in this kind of greenish color. So you can see that the microtubules tend to be internal in the cells, and the actin fibers tend to be near the membrane. The centrosome is like I said, a hub of microtubules. It's kind of a center to that. Microtubules radiate out from there. They also kind of branch off and go in different directions, but these are the things that we use to pull the chromosomes apart in animal cells, forms that cytoskeletal spindle structure that grabs a hold of the chromosomes and pulls them apart. In plant cells, we just attach the microtubules to the opposite cell side, and the cell wall provides that kind of anchor. In the case of animal cells, we use these centrosomes as those. So typically a cell has one, but when it's getting ready to go through mitosis, it makes a second one, and it uses the two of them to make that spindle, and then each new daughter cell has one. Flagellum and cilia, these are parts that stick out of the cell that can move around. They're largely just membrane-covered bundles of cytoskeleton, with a lot of times with motor proteins in them so that they can move around. We kind of mentioned that before with motor proteins, but you don't need to worry too much about these. They do have kind of an organized anchor at the bottom called a basal body that kind of attaches to the rest of the cytoskeleton so that the flagellum or the cilia has like some leverage as it waves. So those are all the cell parts we really need to worry about, and that's really all the detail we need about them. This is just a place where things happen. It's not the important thing. The important thing is what cells can do, not what they have. So you'll see in the next few lectures how we kind of bring some of these things together to make more complicated things happen. So hang in there, you're doing great.